So today you are here for the QAAFI Science Seminar with Jiahua Shi, and I will introduce her in just a moment. But first, Jiahua, do you just want to move forward for me, please? My name is Heather Smythe, if you don't know who I am, and I am Professorial Research Fellow in the Centre of Nutrition and Food Science at QAAFI, and I'm going to be facilitating the session today and taking your Q&A. Next slide, please, Jiahua. Before I begin, I'd like to acknowledge country, and the University of Queensland acknowledge the traditional owners and their custodianship of the lands on which we meet. And we pay our respects to their ancestors and their descendants who continue cultural and spiritual connections to country. And for the land I am on today, and most of us at UQ at the St Lucia campus, that's the Yuggera and the Turrbal people. And we recognise their valuable contributions to Australia and to global society. So today, just some housekeeping. The seminar is scheduled from 12 till 1. So thanks for sticking around. Feel free to eat your lunch. At the end of the seminar, we will have a time for some Q&A, which I can facilitate. Please use the Q&A tab at the bottom of Zoom here. You'll see a Q&A tab to ask your questions. Please don't use the chat function, but Q&A will allow myself and also Megan in the background to manage those questions coming through and ensure everyone gets an answer. Thanks, Jiahua. So now to introduce our speaker today is Dr. Jiahua Shi. Jiahua has been working with me. I've had the pleasure of having her work with me. She's based at the Elkhorn Laboratory within CNAFS and has been with me for, more than 12 months now, Jiahua. So Jiahua Shi is funded through the Food and Beverage Accelerator, and she's a Senior Research Fellow in the Centre of Nutrition and Food Science, as I mentioned. She's a strong foundation in fermentation, food science, analytical chemistry particularly, and sustainable food systems. And her research helps address critical industry challenges, particularly around waste reduction, upcycling of processing residues, development of novel food ingredients, and improvement of nutritional profiles in food and beverage products. She works closely with industry partners to deliver innovative and commercial impacts. So she has a number of industry partners, and she can't talk about all of those industry partners today because there's some IP involved, but she will be able to speak to you about some of the other research that she's been doing on the side. So just moving forward, Jiahua. And from waste to taste, I'll hand over to you to introduce your topic today and a little bit more about yourself. Thanks, Jiahua.
Good afternoon, everyone. Thank you for joining today's QAAFI Science Seminar. Appreciate Heather for the introduction and facilitating. My name is Jiahua Shi. I'm a Senior Research Fellow based at CNAFS at QAAFI. I joined UQ actually in January 2024. Gee, it has been 18 months. But still, I feel relatively new here. And I really appreciate this opportunity to share more information about my research to QAAFI colleagues and a broader audience. Before I start, I too acknowledge the traditional owners and their custodianship of the lands on which we meet today and pay my respect to their ancestors and their descendants. Well, a bit background of me. I got my PhD from Nanyang Technological University in Singapore in 2015. Then I worked for 4 years at NTU Food Technology Center, as well as the APAC Center for Sustainable Development in Agriculture and Fishery sectors in Singapore. I've also worked 4 years at University of Wollongong before I joined UQ last year. So in UQ, my fellowship is based at QAAFI through FaBA program. FaBA is the Australia's Food and Beverage Accelerator. So it is a national initiative supported by the Australian government, industry partners, UQ, QAT, and UniSQ. The whole idea is to help accelerate innovation in Australia's food and beverage sectors. So essentially to translate research into real-world impact. Since joining UQ last year, I've been working closely with Professor Heather Smythe and our industry project teams. Which now includes around 7 senior research assistants and technicians. You would say me like posting the LinkedIn recruiting the advertisement a lot, and we are having several casual staff as well. So together we have with head Professor Heather Smiles, we've attracted around $6.2 million since last year in funding to drive several major collaborative projects. We have secured support from FaBA, GRDC, industry partners, the main ones including MyPlantCo and Bundaberg Group Drinks. I've also secured the FaBA IP voucher scheme and also the CSRO voucher scheme. These projects are very industry focused, so I won't go into too much details today for IP reasons, but they have formed the backbone of my applied research over the past 18 months. Parallel to that, I've also developing my own strategic research team. So currently we have 1 PhD student, 2 master students, and 2 research assistants, where our main focus is on waste conversion and creating the value-added ingredients or products through bioprocessing. So for today's talk, I'll take you through 4 key parts. First, a brief look at food waste, the background and impact. And then I'll explain my research focus and the concept of waste to taste. Then I'll share a few of the current projects we are working on. And finally, I'll wrap up with future directions and where I see this area heading. So let's get started with the big picture. Food waste and byproducts. They are a global challenge that touches every part of agri-food chain. By 2050, the world's population is expected to reach 10 billion. And more than 800 million people are actually going hungry every day. So however, every year, roughly 1/3 of all the food produced for human consumption is lost or wasted. So that's about 1.3 billion tons globally. And it's not just a moral or environmental issues. It's a massive loss of nutrients, energy, and economic potential. So a large portion of this waste does not come from spoiled food, but from the surplus produce that never even makes it to the market. And they are perfectly edible fruits or vegetables. They are often rejected simply because they are the wrong size, slightly mishappen, or just don't fit the ideal appearance that the retailers want. And then further along the chain, our food processing industry generates significant byproducts. Thinking about brewers' spec grain, for example, from the beer production, or the seed or peels residues from avocado processing. So these are the streams that are incredibly rich in nutrients like proteins, fibers, bioactives, but they are typically underused or just discarded. So instead of seeing these materials as waste, I see them as resources, actually, the ingredients waiting to be rediscovered. So that's where the idea of a circular value-add food system comes. So by developing these smart processing technologies, we can close the loop, transforming this surplus and byproduct streams into the premium food and beverage or ingredient innovation. And that's a philosophy behind my research program, turning the waste into taste, and also aligning with the sustainability with innovation. Let's take a moment to understand the scale of the food waste problem here actually in Australia. Every year, Australia produces 7.6 million tons of food waste. That's roughly more than 300 kilograms per person. Well, even more alarming is that around 70% of this waste is actually edible. So it's really like when in China, we're striving for food and there's always food shortage. So my heart breaks when I hear, well, 70% of them actually are just being edible, but being wasted. So in other words, they are not scraps or pills. It's good food that haven't been eaten. And economically, this waste costs us an estimated 36.6 billion every year. So which includes around 19 billion just from the households. And on average, that's over like 2,000 per household. Essentially, money is just being thrown away. So from an environmental perspective, we are wasting more than 25 million hectares of land to grow and manufacture food that end up in the bin. So that's more land than the whole of Southeast Queensland combined, or maybe the whole area of Victoria, just to grow food that ends up actually into the waste. And there's also consumers, this also consumes that to 2,600 gigaliter of water, which is equivalent to about 286 liters per person per day, and generates 17.5 million tons of CO2. So its reach is roughly 3% of Australians' annual emission just from the food that we never eat. So here's the striking part. Around 2.6 billion kilograms of food never leaves the farm. That's nearly 1/3 of what is being produced and rejected simply because they don't meet the beauty standards set up by the supermarkets, the wrong size, the shape, or the color. And they are perfectly good produce, but not pretty enough for the retail shelf. So when you talk about ugly food, it's not really ugly at all. It's a symptom of a system that value uniformity more and over the sustainability. So this is exactly where my research begins. Just asking how we can take this surplus and those byproducts and reimagine them as the valuable resource rather than waste. So this brings us to the central theme of my work, what I call waste to taste. So the idea is quite simple but powerful. So we are taking what is currently seen as waste in the agri-food system, whether it's surplus produce, byproducts, or the processing residues, and turning them into something of value through the smart processing. The first step is to reduce the waste. That means we find the products used from the surplus or the byproducts that would otherwise be discarded. So instead of sending them to landfill or the low value use such as animal feed, we reimagine them as the raw material for functional food ingredients. The second is to stabilize the nutrients. Many of these byproducts, they are actually rich in protein, fiber, antioxidants, bioactives. So they are often not stable, or they can be degraded quite quickly. So through bioprocessing technologies, for example, fermentation, extraction, encapsulation, we can preserve these nutrients, improve their functionality, and make them easier to integrate into the food system. The third step is the deliver of new application. So once we've established and enhanced these materials, we can develop innovative end products, things like high protein snacks, functional beverage, spreads, or even natural preservatives or coating. So in other words, we are not just recycling the waste, we are redesigning them into premium market-ready ingredients and products. And finally, the bigger vision to position Queensland as a leader in this circular food system. So we have the world-class agriculture, very strong in research capacity, and we have an industry that is increasing and open to innovation. So by connecting all these dot points, Queensland has a real opportunity to lead Australia and even in the region in sustainable value-added food production. So waste to taste, I don't think is not just a catchy phrase. It's more of a framework for me that connects environmental sustainability, nutrition preservation, and also the economic opportunity. So that's a big picture, turning waste into value through the smart processing. To make this idea more tangible, I'll start with one of my favorite example, avocado. It's a perfect case study of how we can transform off-grade fruits and byproducts into something functional and premium. Well, when I resumed my research in food science last year, I was asking myself, what do I want to work with? What is a food or food source that is being wasted and not studied too much? They are limited or challenging in industry and with great potential and with great nutrients as well, and developing interests for global market. And to be honest, I was wondering at each single shelf, each single lane of Coles and Woolies and all the fruit markets near my house, and starting to look at each fruit and vegetables, each food products. And there are two successes. I think that two gains, I think, from this experience. First one is I do grocery shopping really quickly now because I'm so familiar with the location of each item. Second is that I find the most suitable research target, which is avocado. So Australia, we produce a large amount of avocados every year. For example, we produced 115,000 tons of avocado in 2023, and we are ranking number 17 in the whole world. Australian Hess and Shepherd are the two main varieties of the fruit. Hess basically occupies most of the market, reaching about 80% of the time throughout the year. Well, Australia's avocado production is growing rapidly, but we are also facing challenge of oversupply. So only around 10% of our current Australian avocados are being exported currently. So we are playing quite small in the exporting markets. And that means 90% of the produce are served domestically for a quite small population of the whole Australia. So as the industry expands, we are also saying, growing volumes of surplus and off-grade fruit. So these are the perfectly edible, but rejected because of the appearance, size, or the minor surface defects. So on the left, this is a picture reported by ABC News in 2022, which happened in North Queensland. So the oversupply results in these pretty good avocados. They are not even the ugly ones. So they are being left behind on the farm due to the high, transportation, logistics, distribution, all these costs. So these avocados are being discarded by the growers. The current as well for the non-edible avocado waste. The current avocado application in the market in Australia is quite limited. So majority is for direct consumption by consumers. A small portion of them go to guacamole, and an even more smaller portion goes to avocado oil. So there are some of the problems in the industry production of avocados because of the oxidation, so including the enzymatic browning, lipid oxidation, so which directly affects the color, the flavor, and shelf life. So during the avocado processing, for example, avocado oil, they generate significant residues, including the peel, the seed, and the pulp, so which still contain a wealth of nutrients and bioactive compounds. And I have one PhD working on avocado project. So before we proceed to transform the avocado waste, we wanted to understand the nutritional value of avocado in Australia. So this is a brief introduction of Australian health, the macro and macronutrients according to FaBA system. So for the edible part, around 70% is actually moisture. And then fat accounts for 13 of the total mass, which is the highest part except for the moisture, of course. And 7.5 of them, they are dietary fiber. They are very good for digestion. And the proteins and ash, they account for 1.6 and 1.7% respectively. It also contains vitamins, not only water soluble, but also the fat soluble ones, which is really hard to say actually in fruits. That means it's a good source for most essential vitamins for vegetarians and vegans. So at the same time, avocado is also full of minerals. They also have other nutrients like carotenoids, organic acid, and phenolic compounds. However, there are no quantitative reports of peel and seed. This is the first problem we feel, because they are the large portion as well, accounting for roughly 30% of the whole fruit. And secondly, the latest data from the FaBA system, at least the FaBA website, is from 2014. And while the earliest can date back to 1988. So many years has been passed. 10 years has been passed even for the latest data they are listing. And I believe we need more recent and accurate data for these beautiful avocados. And in this past 8 months, so my PhD student, Macro Wintel Zhou, he has been working hard to get better knowledge of Australian has avocado. The detailed quantitative analysis of pulp, peel, and seed across different seasons as well. So we have obtained data for proximities, including moisture, ice, protein, fat, and fiber. And we also got data for vitamins, organic acids, amino acids, carotenoids with HPLC, and sugars with SES, the ion chromatography system. We've got data for fatty acids and volatile compounds with GCMS. We've also tested the trace elements with SAPMS, as well as we did open profiling with HPLC and GCMS. So we have found that avocado peel, pulp, and seed, they are all rich in valuable nutrients and bioactives. So the peel, they contain high levels of polyphenols, flavonoids, and carotenoids with strong antioxidant capacity. And then the pulp is abundant in healthy monounsaturated fatty acids, vitamin E and vitamin C, and potassium, while the seed is concentrated source of dietary fiber, phenolic compounds, and natural antioxidants that contributes to antimicrobial and anti-inflammatory properties. So we have been collecting avocados at different seasons, and we'll finalize the 4 seasons analysis this December. So I didn't put detailed data here because they are being prepared for publication. But stay tuned next year, hopefully, that we will have the paper ready. These nutrients and bioactive compounds from the surplus avocados or byproducts, they are actually a hidden treasure. So instead of discarding them, our work aims to convert these waste streams into functional extracts that can be applied in food, beverage, or even cosmetics, pharmaceutics formulations. So essentially, we are rethinking the avocado waste as a biorefinery resource, extracting its full potential and the commercial potential. To achieve that, we are applying a combination of extraction and encapsulation technology. The extraction step allows us to isolate and concentrate the bioactive from the peel, the seed, and pulp. And then through encapsulation, we stabilize these compounds, protecting them from the oxidation and degradation, and then improving their dispersibility in different product systems. So the result is a set of stable antioxidant and antimicrobial formulations, which can be integrated into various applications. Well, we are still quite early stage of extraction and encapsulation, but we've found a very interesting thing, which is barely starting in avocados. We've found several papers from the whole world. This is about oleosome, which is large quantity in avocado pulp, or we can call it oil body or lipid bodies. So as you see from the picture, oleosome is a kind of micro particle naturally exist in the plant tissues. which is the richest storage structure for the plant oil. And the structure is like triglyceride core surrounded by a phospholipid membrane with some proteins embedded in. So the oleosome is a natural emulsifier. They can be easily extracted. In the meantime, the naturally formed phospholipid protein membrane, they lead to the enhanced environmental resistance and stability. So we have been trying different methods to extract and identify the oleosome. Further research is needed to verify the specific results. And once we finalize an efficient way to extract avocado oleosome and testify its stability, we will further investigate the potential to use it as emulsifier to encapsulate the bionutrients in avocado. So the whole philosophy, the whole idea is to biotransform avocado waste using 100% of avocado and to produce a value-added products. So the second example I would like to share is the valorization of brewer's spec grain or BSG through the solid state fermentation. So I have two master students. They have been working for 7 months in my group and co-supervised by Professor Mark Turner and Dr. Yuwei Xiang at Axis. So this project is collaborated with the 4X Brewery in Milton, which is really convenient for us. And they are having the in-kind support for BSG fortnightly, which we really appreciate their support. So BSG is the major byproduct of beer brewing industry. And they are representing around 85% of the total byproducts. So it's lignocellulosic material. They are rich in fiber, in protein and prebiotics. So particularly in the arabinoxylanes and beta-glucans. So despite its nutritional potential, BSG is still underutilized. Well, a small portion of the 4X Brewery, that goes to the very unique Australian spread, the Vegemite, which by all means, and no offense at all to Australians, that I don't know how to enjoy it yet after 7 years in Australia. So this is the unique capability of 4X, but most of the breweries, they either send BSG to animal feed or to landfill because they are very bulky, they're wet, they're not stable. So we were doing sampling like early in the morning, and when the 4X staff, they collected the BSG, which is freshly made, is super hot. And then we use the SK to transport to Long Pocket campus. We have to freeze them really well and freeze dry them like quickly, because otherwise after a few hours, you can feel the aroma starts to change a little bit. And after 2 to 3 days, they cannot stay the original format at all. So they are very tricky to be processed without a good technology. So a lot of the majority of them, they are being just go to landfill or animal feed. So this is not only a waste of nutrients, but also a loss of opportunity to create the value-added products. So what we are doing is reframing the BSG as functional food and ingredients. So with the right processing, particularly fermentation, we can enhance the nutritional quality, we can improve its flavor, texture, and we can open up the new applications in human food system. So to valorize the BSG, we are using biological pretreatment under mild conditions and makes the process not only efficient, but also environmentally sustainable. There's no harsh chemical or energy-intensive steps involved. We also apply solid-state fermentation using the filamentous fungi, such as the Aspergillus oryza and the Rhizopus oligosporus. So these species are food grade. They are traditionally used in Asian fermented food, such as soy sauce and tempeh. making them safe, well suited for food application. So all the fermentation work here in my group, they are food grade. So all this makes us the capability to produce further like food ingredients we can connect it to, for example, follow up sensory because they are food grade. So for these two fungi we were using, their mesocyl network, they form a large surface area that can physically penetrate deep into the BSG matrix. So they can effectively break down the complex structures and they can release the bulk nutrients. So through the solid state fermentation, we can enhance the protein digestibility by breaking down the cell wall polysaccharides and releasing the bulk nutrients. And at the same time, the fermentation, they boost the prebiotic activity thanks to the production of xalanase and beta-glucanase. So these are the particularly hydrolyzed complex fiber into the bioactive oligosaccharide, and they can support the benefit for the gut mycobacteria. So this fermented BSG, they can also develop rich aroma and color profile. So all of these making it very suitable for food application, such as high protein snacks, bakery products, or even functional beverages. And the students has been working hard to perform the strain selection, fermentation parameters, optimization, including the, for example, the incubation temperature, substrate moisture, the humidity to be in the environment, and the fermentation length, the sampling intervals, inoculation amount, and also the tests of co-cultures mix and match with each other. So I'm very proud to share that our students have successfully detected and characterized these key metabolites. So using HPLC, they've confirmed the presence of AXOX and XOS, which are the major prebiotic components. And on the protein side, they have been used SDS page and proteomics analysis via the LCMS Orbitrap. So they map the change in protein profiles before and after fermentation and at different time points. So this provides direct evidence of the enzymatic modification and also the nutrient enhancement with the BSG matrix. So I didn't put detailed data here as the seminar will be for public access, but the students are going to have the master project final presentation end of this month. And if you are interested, just feel free to reach out and I can share the details, the time and location. So altogether, these findings, they strengthen the case for using solid state fermentation as sustainable. They are the data-driven approach to upgrade the industrial byproducts and also turning them into high-value functional food ingredients. They are applicable in a lot of the food products which we can imagine. And this is one of the most exciting development in our group. So this is the new food grade bioreactor, which is currently unique within UQ. They are properly supported by FaBA and Idea Bio, which is led by Professor Esteban Meselin at AIBN. It has been purpose built to operate under food grade conditions. So this allows us to conduct liquid fermentation with precise control over temperature, humidity, and aeration, agitation. So everything is under control and monitored. So what makes this quite special is that it's in our food grade lab. All the other bioreactors in UQ, they are located more in PC2 lab. So what makes our bioreactor special is that it can integrate with the downstream analysis, which I mentioned earlier. So we can take samples directly from the reactor. We can run the biochemical, microbial, or sensory testing in-house. So this gives us complete view of how the fermentation changed the ingredients chemistry, how do they change the flavor, and also the consumer perception. It's also designed as an industry-ready platform, so they are ideal for pilot scale trials, for IP development, and they can bridge the lab scale discovery with the commercial application. So I have one research assistant working on the production of plant-based cheese currently. So with these bioreactors, and hopefully we can develop some market-ready prototypes soon. In my vision, these food-grade bioreactors, they are helping positioning UQ and QAAFI as leaders in the food-grade fermentation and the circular bioprocess innovation. For the technical message, as we wrap up, I want to leave you with the core message behind my work. So each tons of surplus or the produce or industry byproduct, they are not waste. I treat it as more an opportunity. So through the bioprocessing, we can unlock the new nutritional, functional, and commercial value. And also through innovation in this space, they don't just reduce waste. It actively drives sustainability and creates premium markets for Australian producers. So looking ahead, my team is expanding the solid state fermentation to other underutilized crops. We want to try with you, for example, the oat husk from our oat projects, the sorghum, the fruits, pomace, and we can integrate with AI or data-driven analytics to monitor and optimize the fermentation processes. And we're also building strong industry partnerships and moving towards a pilot or commercial scale application. So we want to bridge the gap between research discovery and market reality. So ultimately, I believe Queensland UQ, we can lead the waste to taste revolution, showing how the circular food innovation can be both sustainable and profitable. And at last, I would like to acknowledge a lot of people. I know it's a long list, but in the past 18 months, I really appreciate with multiple success, and it cannot be done without the help of everyone. So I really appreciate the QAAFI leader team, especially Professor Matthew and Yasmina. They have been supporting every single step of my career decision, further direction, and when we have problems, we kind of solve them. When we have success, we cheer them up. And to Professor Heather Smythe, which I need to highlight that all this success won't be happen without Heather's help. I guess this is a quite difficult step when you have career disruption, you're having different change in roles in different universities, especially for international relocation. And Heather has been really great to actually one of the best supervisors you would ever expect to show you how to communicate with industry partners. They showcase and gave you the connections with them to initiate these projects, which won't be happening for a new employee at all with this major grant secure. And so I really appreciate it. Don't want to be too emotional. And also I would like to share Michael for being, helping with all the analytical, being the panel for my PhD student, and also Alice and Chris. They are from different centers, but they have been helping so much for the avocado, detection, linking us to the avocado team, to the avocado farm, even to make our thinking more realistic. Because in reality, we are, for example, buying avocados from Woolies, but how do we pick them? This experience from the farmers, from the growers, they are the most valuable things that you will not learn from the literature. And also my students, Yuntao, for the hard work, Wenkang for the hard work in maintaining the bioreactor. I also thank QAAFI, the BD team, the marketing and communication, admin team, finance, operation, HSF. I've been bothering all of you enough, I guess, to purchasing all the things, processing all the things, and really appreciate everyone's help. And because I'm based at FaBA, I really appreciate the FaBA executive team, Chris and Sue and Kestra. They have been supporting in every aspect, either from financial support or the project discussion. They have been so supportive along the way. And also Professor Jason Stokes, we have been having, meetings every 2 months to get me in the correct track. And also the collaborators from AIBN, Professor Esteban and Dr. Axia and Huadong, they have been helping so much with the bioreactor. Before that, we have no idea how to set up here in our home building because we don't have it yet. And they are very well established within AIBN. And they are so generous and so willing to collaborate and sharing their experience and help us build establish the platform. So I really appreciate. For the whole QMAP team, they have been helping with all this analytical. So training of the students is not just sending samples to them. We are not doing sending sample as a service. What we do is train the students with every step being the hands-on done by himself and all the education, the knowledge from the QMAP team. I would also like to acknowledge Dr. Ran Zhang and Dr. Miao Miao Wu, we have been collaborating on the avocado extraction together with nanotechnology for the shelf life extension on fresh cuts, which I didn't share too much today. From ACFIS, I really appreciate Professor Mark Turner, Dr. Yu Wei Xiang. They have been helping so much for the fermentation work, and also Dr. Alberto and Bernadine. They have been helping my students in different ways, and also Miss Xintong and Mr. Ziwei Guo. They are my 2 master students who have been working so hard and very impressive progress as a master student with only short time. And again, at last, I would like to acknowledge FaBA, GRDC, and all the industry partners for their generous financial support and also the in-kind support from for our projects. And I guess this is the last step. Feel free. So today I didn't share too much about the industry projects or the data because this will go on YouTube, I guess. So all of them are a showcase of our capability and what we are doing now and in our vision. So feel free to reach out if you have ideas, if you think, no, you're doing it wrong, or you can do it better. So just feel free to reach out, and I'm so happy to collaborate with more and more people. And I'll head back to Heather for facilitate. Thank you.
Thanks, Jiahua. That was fantastic. And it's been a delight having you on board. You're an amazing collaborator. And our friends at AGFIS and AIBN have been really great, and you've worked with them really well. So I'm grateful for that as well. There are a few questions in the chat, and I'll go to those in just a second. I just wanted to, I guess, ask you about, it is interesting that we have this bioreactor now that's in a food grade lab. But it is not a PC2 lab, and I suppose we've had a bit of a chat about that. The need for having both the PC2 and food grade lab in combination for this. What are your thoughts about that? What do you think that might achieve for us?
Yeah, I think so. For example, Professor Esteban and Professor Mark Turner, they have been establishing many years for fermentation. Especially for the precision fermentation, genetic engineering, and for targeted and untargeted, for example, small molecules. They can be the innovative ingredients. But I think they are working on PC2 lab and all this follow-up assessment. There is still, for example, a debate. If the genetic modified organism can we take them for the food application? I think recently Australia has approved that the extracts from the GMOs to be used for food application, but still the directly products from the bioreactor, if we want to do sensory, I still feel it's a challenge. So this is we are not saying we're competing, we are collaborating in every single way that we can think of. This is a capability and beauty here in Long Pocket Campus in alcohol building, that with your food kitchen, sensory, and we can on-site directly after fermentation, we can do all this follow-up analysis. I think this is by all means that we can expand from the lab scale to pilot scale and to industry scale. So it's complementary to each other, I'll say.
Yeah, I'd agree with that. Absolutely. And I think we're a bit behind industry in that they have food grade PC2 labs for doing their kind of GMO work. But at universities, when we ask the question, we get a few raised eyebrows. You can't eat anything from PC2. Well, you can if it's a food grade PC2 lab.
Yeah.
Anyway, I'll look at these questions here, Jiahua, because we've got a few coming through now. So we've got Doris Wong is asking some questions. She said, your project values align perfectly with my interest, as I'm also interested about sustainability and reducing food waste. And she's talking about, and we have previously initiated a project creating an online grocery platform utilising ugly produce and exploring upcycling of food, particularly avocado seeds, into extruded snacks. So she's an active volunteer with Oz Harvest and Fair Share, and I have rescued large amounts of ugly produce across Brisbane. Through this experience, I've seen the wide range of conditions in which the ingredients arrive, I guess the waste ingredients, and have cooked meals using these products. How do you maintain ingredient quality and minimise contamination risks while retaining the nutritional and functional properties and, I guess, sensory, from my perspective, of those byproducts?
Thank you. I think, this is quite crucial that sometimes we treat it as that's why in reality, there's still a long process to go between your lab production to the pilot scale or even to the commercial scale. I think in industry, they tend to do it very quickly, saying, well, this is food grade, we're doing this extraction and should be safe. But in our lab, if we want to do all this follow-up tests, everything for example, the microbiology, they need to be tested, all the shelf life needs to be tested, and minimizing the contamination risk is just to control all the conditions really carefully, but just to think about your whole process, the design, and also and even need to consider from the lab scale, they will be different from the pilot scale. This is one of our industry projects where we have been having difficulties, because in lab scale, we can do a lot of these procedures that is very easily, but it's not realistic to apply them into the industry. So I think contamination risk, they can be treated with quite, for example, the HPP, high pressure processing, all these facilities, but just to keep in mind what is the feasibility when you scale up. For us, for the avocado job, we are very still initial stage to extract the oleosome and doing for the encapsulation. But later on, we need to think about all these risks, being contaminated or being not stable. So how are they going to be changed after 2 days? Are they even changing to other ingredients or compounds? So these are all the things you need to test before releasing the products.
Yeah, definitely. And you have to handle that waste, I guess, according to what you're going to use it for, isn't it? And what you need. kind of quality you need. She's also asking, what different approaches or technologies have you found most effective in keeping bioprocessing of byproducts economically viable without compromising quality, considering your goal is scaling those bioprocesses? Yeah.
Yes. That's why I think when we call it cellular economy, like food system, and sustainability is a buzzword that everyone is talking about. And to be honest, I work with Technical University of Denmark, the Danish tech, and I've been trained for sustainability assessment, doing the LCA, all these quantitative and qualitative assessment of sustainability. A lot of the food waste conversion, they are actually maybe just suiting one of the perspective. Because for the sustainability, we have, economic sustainability, environmental, and social. So a lot of the things we're considering now, maybe more from the economical part. And for the environmental, how to balance them, how to get the best point is always very tricky. So what we are using now currently is that to think ahead, what in the industry application. For example, if we can use water, we don't use other chemicals. If we can use lower temperature, we don't use high temperature. So, for example, microwave, they can extract this compound so easily, but they are just expensive or not applicable in super large scale. So this, when we consider is that how feasible it is to change for the industry, as saving their water, saving their electricity is everything before you design your experiment. It's not always like, oh, I'm extracting this. We can publish paper, a very good paper saying this is the best way we can extract it. But again, you are spending like $1,000 to extract some proteins, and which by all means, there's no point of doing that. So for us, we are just focusing on using the limited input and achieve the most output. But you're right. Bioprocessing and the biotransformation can be expensive. The things that how to balance the input and output and take the sustainability into consideration, not saying we're rescuing waste and sustainable. It's all quantitative, not just by saying conversion.
Yeah, definitely. Yeah. And not having to invest in very expensive equipment at upscale. That's tricky. The next question is around skimflation, where companies reduce ingredient quality, such as replacing oats with rice puffs in muesli bars. Could such substitutions be considered a form of resource optimization, or do they risk undermining consumer trust and nutritional value, especially if driven more by cost cutting than sustainability? So what are your thoughts about that comment, Jiahua?
Yeah,
so I guess companies calling it, we're being sustainable, but in actual fact, they're just, maybe reducing nutrition by replacing those ingredients instead.
Yeah. I guess, I think some of the companies, I feel they are more for marketing, not for, the real benefits or so I think for industry is not it. The thing we find it different is that they want to balance more for marketing and also the consumer perception. How do they accept the product more instead of, marketing in this side, but they are compromising the other side. I think that this is a very fair point to consider, I think.
Yeah, exactly. Yeah. Michael Netzels just makes a couple of comments. Great presentation. And he's saying that avocados can also be a really valuable dietary source of folate. That's great.
Yes.
Do we have any waste numbers for strawberries in terms of waste in Queensland? Do you know any of the numbers of, kilograms, tons of strawberries that are wasted each year? Not sure.
I have no idea for the strawberries. I think two of the students they are collaborating with the strawberry farms. So they are trying to identify the bionutrients in the strawberries and also the sensory, correlation. I think that you have more experience with strawberries.
For sure. It really varies from year to year. Do you remember several years ago there was
Yeah.
some student made up a story about a needle being found in strawberry packets, and so much was wasted that year. But it also depends on the rain in the season, I think, in terms of how much strawberry waste there is. We're nearly out of time, Jiahua. I'll
Yeah.
ask another question. Is there time constraint or pressure from when your BSG, it's BSG, isn't it, needs to be
Yeah.
collected before processing? So how soon before you process it do you need to collect it?
And for us, I guess for us is that we communicate with the Forex brewery staff and arrange like maybe 1 week before their collection time. And once they collect it, we told them to just put in the fridge, the 4 degree, to minimize all the enzyme, digestion, all the enzyme activities. And then we bring esky with, ice blocks into it, and just 20 minutes drive back. Whereas at UQ, we just process on the day. So because we have been experiencing, for example, after 4 hours, because they're super hot, if the fresh ones, they will be, changing a lot of the aroma and the color, and also the texture, this become, a little bit more sticky. So for the time constraint and pressure, I think we appreciate that we are so near to the Forex brewery, so this is not tricky for us to think about this yet. But for the industry scale up, I think it's better always to have the brewery and have a processing plant just next door or just within the plant, so they can process on the spot and do all the extraction and further processing. This is a dream for all these, large scale, because the logistics and also the human labor, there will be such a great cost. I think usually this logistics cost most portion for this waste rescuing, because there are like thousands of tons every year, and just from one single brewery. So we'll be thinking that if this is successful, we will be, having, I don't know how scale up we can be successful, but we have a, pilot scale, maybe just next door within the brewery.
And I think that's what they wanted, wasn't it? They were looking for a solution they can implement rather than trying to offload their BSG to somebody else.
Exactly.
I saw a really interesting presentation about using BSG to make biofilms, actually, to use in product packaging. You've probably seen a few papers about that. Jiahua, that was something I saw at the conference when I was away. We do have a couple of other questions, but we're just about out of time. So I'm going to, again, thank you very much for your presentation. It was fantastic, Jiahua. Great to hear from you.
Thank you, everyone.
Such an enthusiastic presenter. If you just go to the next slide, I'm going to give a very quick and dirty plug for TropAg, as I should. Coming up on the 11th to 13th of November, please come along. It's a great way to connect with your peers working in the area. We've got 3 themes: future foods, growing agriculture, and also climate and sustainability. So if you're not from UQ and from industry, please come along. We'd love to have you there and make some great connections. And if you are from UQ or QAAFI, I'd also love to see you there if you're able to make it this year. So that'd be great. Next slide, please, Jiahua. For anyone who wants to keep up with this seminar series, grab your phones. Here is our seminars webpage. You can sign up for notifications about future seminars in our QAAFI seminar series. Other than that, thank you everyone for coming. You had a great turnout, Jiahua, today. Have a great rest of the day. Thanks everyone for coming.
Thank you, everyone.